Today we will discuss about eukaryotic DNA replication in chromosomal basis of inheritance, Unit 7, Chapter 3 in Botany. So eukaryotic DNA replication. So U means true, karyot means nucleus. In nucleus the DNA is there and it going to divide. The DNA going to replicate, multiply to produce the daughter cells. So the replication starts at a specific site. So in a particular site only the replication starts in the DNA. That site is called origin of replication. So for DNA replication you need the origin of replication. This is the key point. So the origin of replication is the key site, that is the particular site where the replication going to start. And more than one origin of replication is there in eukaryotes. So eukaryotes means true nucleus. So it has so many chromosomes. So each and every chromosome there is so many ORC, that is origin of replication. So for example, in yeast you have 400 origin of replication. So this they may ask in 1 mark. Yeast, Saccharomyces cerevisiae. You have 400 origin of replication. And this that is the DNA replication in eukaryotes will assembled as a pre-replication complex. Pre-replication complex which can be written as pre-RC, that is replication step. And it contains, pre-RC contains 14 different, that is 14 different proteins. Okay, 14 different proteins and they are grouped in 6 proteins. They are in each group 6 proteins are there. Okay, 6 proteins are there and it is initiated by the origin of recognition, that is origin of recognition complex known as ORC. Okay, and this that is origin of replication in yeast has ARS sites, autonomous replicating sequence. Autonomous means automatically it starts its work when it attains a time for replication, that is the DNA attains the time for replication. So that time it starts replicating autonomously. Nobody will tell to do this work, to do that work. It automatically starts when the time comes, it starts replicating. That is called autonomously replicating sequence is there in the ORC complex. Okay, that acting as an initiator, inducing automatically to do the work. In yeast, the ORC, that is origin of replicating complex, has identified a protein complex. Okay, it is a protein complex which joins directly with ARS element. So first what are in this slide, what are you have to remember is origin of replication, that is the site for replication. And more than one origin of replication site is there in the eukaryote. So example is Saccharomyces cerevisiae, that is yeast. It has 400 origin of replication. So DNA replication in eukaryotes, that is pre-replication complex, pre-RC, it contains 14 different proteins and they are grouped in 6 protein groups. Okay, 6 protein groups. Okay, and they acting as an initiator, that is initiating the replication, and due to this they autonomously, that is ARS, autonomously replicating sequence are formed and they do the work automatically. They start the replication and this protein complex, that is ORC, identify the protein complex which is joined directly to ARS. Okay. Okay. Next, the replication fork site. So this is a replication fork. It looks like a fork. Okay. So in that site, there is here that this is the parental stand and this side one parent and this side one parent stand is separated. Okay, the separation of parental strand and one daughter strand. So this is one, this side it is daughter strand and this side it is a daughter strand. So one parent strand and one daughter strand is formed in the replication fork. So there are multiplication, as we had discussed, the yeast contains 400 ORC. Like that, multiplication, replication, so many replication forks are formed in the eukaryotes. Okay, so the helicase, so here the helicase enzyme is that is it starts the, what is the function of helicase means unwinding of the, here the winding takes place. So unwinding of DNA molecule takes place by the helicase enzyme. After the unwinding, here it becomes a supercoiling. So this supercoiling was released by topoisomerase enzyme. Okay, topoisomerase enzyme. And the next one is polymerase enzyme. So the polymerase enzyme forms the RNA primer. Okay, RNA primer in the leading strand and in the lagging strand too. So in the leading strand it is continuous. It starts from 5' to 3' and 3' OH should be free to form the strand. And this side in lagging strand it is discontinuous. You can see the segments of the RNA primer. Okay, it is discontinuous and it is joined by DNA ligase. So it is a paste enzyme. So it joins the DNA fragments which is called Okazaki. These fragments are joined by DNA ligase. And DNA polymerase is acting for the elongation of the strand, leading or lagging strand. So that is the thing. And in polymerase enzyme, okay, the polymerase alpha, that is DNA polymerase alpha, synthesizes short primers of RNA, that is in lagging strand. DNA polymerase D, the main replicating enzyme of the cell nucleus. Okay, DNA polymerase delta, extension, that is DNA strand is extension from in the fork. So in the fork it starts extension. And DNA polymerase beta doesn't have any role of replication in normal DNA function, but removes the incorrect bases from damaged DNA. So this is called repair. As we see the yesterday in the repair, no? That is the repair is repaired by DNA polymerase beta. This they may ask in 1 mark. So DNA polymerase alpha synthesizes the RNA primer. Polymerase delta replicating, that is the main replicating enzyme of DNA nucleus. And polymerase epsilon, extension of the replication fork of DNA. And DNA beta, it doesn't work in the replication of normal DNA function, but it removes the damage in the DNA, and this function is called DNA repair. That was taken place by DNA polymerase beta. So you can see a lagging strand and a leading strand, what we had discussed. Okay, that only. So this is a replication force. What happened? The helicase enzyme is put. Due to that, the release of the strands, parental strands takes place. And the topoisomerase is put here to release the positive supercoiling. And now the DNA polymerase enzyme what it is doing means synthesizing RNA primer, that is with the help of 3'OH, and here also 3'OH, and this leading strand is continuous and this side lagging strand is incontinuous. So this lagging strand incontinuous RNA primer acting as a template for the DNA, and it is joined by DNA ligase enzyme. And these fragments are called as are found out by Okazaki, that is Reiji Okazaki and his colleagues. So they named it as Okazaki fragment. The segments of DNA It is called as Okazaki fragments. They may ask in 2 mark or 3 mark, and this Okazaki fragments are joined by ligase enzyme. Okay. And the same thing here. After that, that is the phosphate bond, everything is formed, and this strand, the one strand is parental strand and the other strand is the new strand. So semi-conservative, that is only one strand is formed. This strand is new and this strand is parental strand. So off only new, off is from the parent. So it is called this DNA replication in eukaryotes. It is an semi-conservative method of replication. And Arabidopsis telomere sequence, that is TTAGGG, thiamine, adenine, guanine sequence. This is the telomere sequence. The plant lacks telomere clock plant meristem cells, produces telomerase enzyme. So, the meristematic cells has an unlimited ability to divide. So, this is the telomere sequence, TTAGG. This was lacked in the telomere clock plant. Due to this, that is the meristematic cells produces telomerase. So, telomerase, due to this unlimited ability to divide. So, it cannot able to, the telomerase, what it does means the meristematic cells won't divide. Meristematic means it goes on dividing and producing new cells. If this enzyme is present, that is telomerase enzyme is present, the meristematic cells won't divide. It does not have the ability to divide. In plants, telomeres do not shrink as in stomatic cells of vertebrates. So, if the telomeres have means the plant cell won't shrink, that is it won't shrink as the vertebrates. But the telomere levels are higher in root tips and seedlings, which has a higher amount of meristematic cells that proliferate the structure like leaves. So, if this sequence is absent, that is this sequence is absent, it cannot able to divide. Okay, this enzyme what it divides means it divides the meristematic cells to divide. Okay, but if it is absent, the sequence is absent means it cannot able to divide. So, the same way the telomerase enzyme, that is it won't do not shrink, okay, as in the vertebrates. So, the roots, seedlings and the leaf tip, what happens, it goes on divide and it produces the new cell. Okay. So, what is the special mechanism which replicates chromosomes ends? After the replication of the chromosome, the enzyme telomerase adds several more repeats of DNA sequence in the telomeres. So, the telomeres use short RNA molecules as a template and add repeated sequence on telomeres. So, the DNA nucleotide polymerization occurs. So, the telomerase, what means it does? It is meristematic. So, it produces repeats of DNA and sequence of telomeres. Due to this, meristematic activity is taken place. That means it acting as an RNA molecule as an template. So, template, and it forms repeat sequence on the telomeres. Due to this, DNA polymerization occurs. So, the energetic of replication, deoxyribonucleotides such as deoxyadenosine triphosphate, that is adenine in DNA, ATP, that is deoxyadenosine triphosphate, deoxyguanosine triphosphate, DGTP, deoxycytosine triphosphate, DCTP, and deoxythymidine, that is thymidine triphosphate. So, they give energy to the synthesize the DNA. So, these are the nucleotides which gives energy for replication, okay, to synthesize new DNA. So, what is the purpose of deoxyribonucleotides? It acts as a substrate. So, substrate means where the function going to attack. So, you are reading, you are the substrate. For what purpose? If you read, you will get a job, okay. Based on your qualification, they will give a job. That is the substrate. The qualification is the substrate for you. Like that, the deoxyribonucleotides are the substrate for DNA replication. And it gives energy for polymerization. Polymerization you have come across in chemistry. The same meaning here also, to produce polymers. So many types of DNA has to be replicate here. Experimental evidences of DNA replication. Taylor's experiment. J. Herbert Taylor, Philip Woods and Walter Hughes demonstrate the semi-conservative replication of DNA in root cells of Visia faba. So what they have done means, they labeled the 3H thymidine radioactive precursor and perform autoradiography. So they have insisted thymidine, which is having radioisotope, that is radioactive material. So they grew that is on the basis the root, that is the root tips are grow. Okay, the root tips are grow the radioactive labeled thymidine. Okay, so the DNA of these cells are incorporated, they produces. Okay, and the outlined labeled chromosome forms the scattered black dots, that is due to the film which they had absorbed in the radioactive or absorbed in the X-rays. In the film strip they found out the thymidine particles. You can see here in the diagram the first generation. The thymidine, that is the chromosomes are treated with, the root tips are treated with thymidine. Okay. In the second generation you have get a labeled thymidine and that is labeled thymidine removed unlabeled in the medium. So it is removed, labeled thymidine, but it is removed in the medium. Okay. And what is in the next generation? You get, that is this one and this one, it is from the thymidine labeled structure, that is thymidine root tip cells are we are getting that, but the others we are getting normal one. Okay. So the root tips which labeled chromosomes were placed in an unlabeled medium. So we don't know what the labeled medium, it contains colchicine. So why the colchicine? To stop the cell division in metaphase. So they use the colchicine. So they take the chromosomes in a radiography plate. And what they observed means, the first generation, so they distributed both the chromatids. Okay. Original one and labeled one. Okay. From the unlabeled one. Okay. And in the second generation, one of the two chromatids are labeled. Okay. And it is acting as a semi-conservative. So now you can see. So in this, the first generation, it is labeled one. One is labeled, another one is the second generation, one is labeled, another one is, this one is unlabeled, this one is labeled. And in the last generation, you have the semi-conservative method, that is one is from the parent type and another one is the new one. So, okay. So this and this is parental type and this one is the new one. So like that, it shows that DNA is a semi-conservative method of replication. So now you understand the DNA replication in this video. If you like the video, give a thumbs up, share and subscribe to Science Easy Tech channel.